Right. So we ready to go?
hmm
All right. Well, hello everyone, and welcome to the presentation for Hans Graham, the scientist who changed microbiology. I am Brian Tickram, and I'm joined by Christian Court. We put together this PowerPoint, and we hope you enjoy.
So, who was Hans Christian Graham? Hans Christian Joachim Graham was a Danish physician and bacteriologist who was born in September 3rd, 1853 in Denmark. And before earning his medical degree in internal medicine and becoming a professor of pharmacology, he first worked as an assistant botanist studying the natural sciences. So, even from when he was younger, he was still kind of learning the minute specific details of botany, which later he earned his medical degree from the University of Copenhagen. So his academic prowess and accomplishments started early on, where he was making indelible contributions to microbiology and as well as science and medicine, such as identifying the correlation of enlarged blood cells, which are macrocytes, as a distinguishable characteristic to the condition pernicious anemia or pernicious anemia, which is when the body doesn't have enough healthy red blood cells to absorb vitamin B12, and we use vitamin B12 as energy source. Um, and later, Graham transitioned from where he was to internal medicine, where he practiced as a resident physician at the Municipal Hospital of Copenhagen.
Okay, so why does Hans Christian Graham matters? Um, we'll get into what he discovered and how they changed the field of microbiology. But not just that, all of science and medicine, even today, we're still using his methods of staining to identify bacteria and help treat patients of their diseases. So he pioneered the Gram stain in 1884. And like I said, he changed how we classify and treat bacterial infections. And he founded the foundation for modern bacteriology and antibiotics, which obviously we're still using today. So, what was known before Graham made his discoveries? So, Graham, as Christian said, started in botany, where he learned about plants and microscopy, and that background helped him understand how stains interact with cells. So, after studying medicine, he worked in hospitals and labs, which led to his big discoveries. So before the 1800s, or rather during the 1800s, microscopes were still improving. There was a few bacteria that had been discovered, but they were very hard to classify. There's no way to distinguish between the different types, and staining techniques were often very basic and unreliable to identify what you were looking at and differentiate between different types of microbes and bacteria that might be under the microscope.
So continuing on to what Brian was just saying, the two different staining methods that we covered in chapter 3. One method was the methylene blue method, just using determine the biochemistry and the morphology, which is like the shape and arrangement of different bacteria. And that was discovered or used in 1876 primarily. Silver nitrate, or the silver stain, was used to identify mainly bacterial motility structures such as pseudopodia, which is like the false feet seen in white blood cells along with amoebas. And then you have the flagella also. And that was mainly popular during the mid to late 19th century. And obviously with the Gram staining method, which is the one we're covering now, which is one of the most famous staining methods, differential staining methods. The acid-fast stain was also another popular staining method, which again, the Gram staining method along with the acid-fast staining method, that stain attaches to specific cell walls or cell structures within the cell wall. And that was used between 1882 or 1883. That was when it was formed. So the Gram stain.
Alright, so Graham's breakthrough in 1884. Um, his stain was, some would say, very elegant. The violet dye stains all bacteria, but alcohol removes it from some, and these are known as gram-negative stains. Others hold on to the violet and appear blue or purple, and those are identified as gram-positive. It was a very simple yet revolutionary way to classify bacteria by their cell wall structure. There was a four step stain method: crystal violet, iodine, alcohol, and safranin. The bacteria were separated, as I said, into gram positive and gram negative, and he published this method while in Berlin during 1884.
So, oh, we gotta fix that, bro. Um, the first four slides, first 1 to 4 to 5 steps are on how to fix the specimen or fix the slide, which we covered again in lab today. But the first step is to prepare 2 slides or 1 slide, depending, one observational slide and then 1 experimental slide, just for the specimen spear. And then we're going to use the inoculation loop, that little, that metal thing that we used. We're going to get a very small smidgen of a sample of the specimen, and then we're going to put that onto the microscopic slide. But before we do that, we're going to add like probably 1 to a few drops of water, and then mix that inoculation soup with the specimen on. And we're going to use that to spread that specimen out onto that slide so that we have a better field of view when it comes to looking under the microscope. So again, step 3, again, just using that inoculations loop, we're going to spread that specimen culture over a circle using a very thin, small diameter, again, so that we're able to, when we zoom into the high power mode, we're able to see that specimen very clearly. And then depending on how much time you have, you're either going to use a heat source or let it air dry. But again, this is all just to fix and stain the specimen. So again, when we use that heat source, we want to make sure that we're not holding that specimen right over the flame, or else we're going to cook it, and then we're not going to be able to see anything under the microscope. So again, step 6 is just reiterating what I just said. You don't want to overheat that specimen. You just want to make sure that you're gently waving that specimen over the heat for probably like a minute, minute and a half, depending on the specific heat. And then we're going to allow that specimen to adhere to the slide, so that when we're staining the method a few times and washing it, that specimen is still going to stick to that slide. So as Brian just mentioned, Gram stain is only really 4 steps. The first step after we stain it is going to be the first step right here, number 7, which is the primary stain. And we're going to be adding that to both specimens, both slides, and it's going to be a crystal violet hue. And what the crystal violet is going to do, it's going to bind to the cell wall of both gram positive and the gram negative cells. So once that's done, we're going to go to step number 8, which is washing that primary stain off, which is again the crystal violet. And we're going to wash it with water, but we're going to make sure that we're not using that water right over the specimen itself. We're just going to allow the water to flow as we hold the slide at an angle. We're going to allow the water to slowly wash through the specimen while still keeping that specimen intact. And then afterwards, we're going to use that iodine or that mordant. It's going to lock that crystal violet only into the gram positive cells only, and we're going to elaborate that a little bit further. But the iodine is to lock that crystal violet in place. After we do that, then we're going to decolorize the stain, which is by using acetone or ethanol, whatever. And what that's going to do is going to, in theory, it's going to wipe and get rid of what is already locked in from that primary stain. For the gram positive, it's going to be that purple hue, like I said. But since that crystal violet is already binded to the cell wall within that bacterial specimen, it's going to again lock. And so we use that decolorization to see if it's going to get washed away. In the gram negative, we'll notice that when we use the acetone or the ethanol, the crystal violet that was once locked into that gram negative is going to be washed away. And then when Hans Graham first was using this method, he would stop it here at this step because he concluded that once he saw that the gram negative didn't have any more of that crystal violet hue, he concluded that that was a gram negative cell. But it wasn't until Carl Weigert, who was a German pathologist, he's the one that added the fifth step, or the fourth step, excuse me, the counter stain, which is that safranin. It's going to be that pink hue, and that is going to bind to that gram negative cell. If we use that gram negative, that counter stain safranin, and we add it to the gram positive, the crystal violet is already binded to the cell walls. So the adding the safranin isn't going to change anything regarding the color of it. But when we add the safranin to the gram negative, we're going to see that the pink hue, that safranin color, is going to bind to the cell walls, hence giving it that, that pink color, solidifying that it is a gram negative cell. So the discovery of the gram stain. So again, this, as Brian mentioned earlier, it was discovered and devised in 1884 with another microbiologist called Friedlander. Hans Graham discovered and devised the gram staining method. But when he first started, he wanted his focus to be studying the bacteria found in pneumonia, because a lot of his patients were succumbing to that disease. And he wanted to use that to differentiate the different kinds of bacteria found in the lungs and other specific tissues regarding the respiratory system.
Right. So how did what Friedlander and Graham did, how did it change microbiology? So Graham's technique gave us microbiologists a tool that's still used today, as we've said before. It helps guide treatment decisions because certain antibiotics only work on certain bacteria, and without it, modern infection treatment as we know it today wouldn't exist, and we wouldn't be able to treat as many illnesses as we are able today because of what Graham did. So he helped identify pathogens much faster, and is the basis for antibiotic treatment. For example, penicillin works better on gram-positive stains. It's still used as a first step in bacterial foundation, and it's still a foundation for modern labs and microbiology. So, what was the broader scientific impact that he had? Beyond microbiology, Graham Stain showed that small differences in cell walls could mean big differences in how organisms function, and this helped shape the way scientists think about all living cells. So, it influenced cell biology, immunology, and pathology, and it led to better classification systems. And it inspired further research into bacterial structure and antibiotics. So his societal impact ranged from tuberculosis to pneumonia. Graham Stand allowed doctors to quickly identify the cause of infections and treat them correctly. Uh, it has helped combat global health crises and reduce death from bacterial infections. It improved disease diagnosis and control, guided antibiotic development, and saved millions of lives through better treatment and enable public health response to bacterial outbreaks.
So after the discovery, again, as Brian just mentioned, that this stain is still even being used today, a few centuries later. So obviously this being used to teach and practice medicine. And as we know that we're always practicing medicine, that nothing is ever solidified because we're always discovering the human body because things are always changing. So Even though the Graham stain was never patented, hence by the name, we always know that Hans Christian Graham was the one that was responsible for devising and discovering the Graham staining method. And since he was so humble about his work, he called it a method that was very defective and imprecise, which is probably another reason why Dr. Carl Weigert, the German pathologist, added that fourth step of adding the safranin to make sure that those false negatives and false positives weren't a regular part or regular practice of the procedure and the method.
So what was the legacy of Hans Christian Graham? Graham's story shows how curiosity and basic science can lead to breakthroughs that touch every corner of medicine and society. His stain obviously continues to protect lives every day. It shows that how even a fluke or a mistake would become a very important method. Graham remained a dedicated teacher and never profited from his discovery. As Christian said, he didn't patent his stain, and he acknowledged his flaws, but he didn't realize how powerful and long-lasting his method would become. That method was a simple method with global impact. It changed how we combat disease and a reminder that even imperfect discoveries can save lives.
Perfect.
For our references, we use the CDC and NIH, as well as our textbook and other biographical sources on grounds such as Britannica and PubMed.
Thank you all for